So our series this year is entitled Animals and Humans. We're focusing our series this year on people's relationship with animals. They serve as our companions, as our friends, as religious figures, as icons of beauty, as sources of food, and clothing, and power, and protection, and knowledge. So we're seeking here a fresh look at the ever-evolving relationship between animals and humans, and ask, among other questions, whether animals are mere biological machines, and what is the ideal way in which humans can interact with other species. So following Dr. Zoghbi's talk today, we'll have a full agenda. We have a full agenda lined up for the spring, including planned talks from Carrie Wolf in Rice's English department, Dr. Tony Wright, who I saw come in and is here somewhere right in front of me, from the University of Texas, Department of Neurobiology and Anatomy. And this is late-breaking news. We will have a talk from C.J. Peters from UTMB, University of Texas Medical Branch, Galveston, who's an expert on the transmission of diseases from animals to humans and may have something to say of relevance to the avian flu question. I'm also delighted to remind you that we have a Bachner lecture lined up for the spring, and that's Dr. Irene Pepperberg, who will be giving the Bachner lecture on the evening of Monday, April 10th. And for those of you who don't know about Dr. Pepperberg, her research with her African gray parrot named Alex is delightful and amazing. What she has taught that bird to do is astonishing and hard to reconcile with many prevailing theories of cognition. And in fact, Alex the parrot is now teaching other parrots things, so it's conceivable that Alex will be eligible for a faculty appointment at Rice sometime in the next few years.
Will he give the lecture?
We're working with Alex's agent. It's possible. His fee is simply unimaginably high. So lastly, before I introduce Huda, let me just note the format. Huda will speak for 50 minutes or so, and then we'll go into 10 minutes of question and answers. We'll have roaming microphones so that the people who will view on the videotape can hear the questions being asked, as well as the rest of us in the room. And then we'll have a wine and cheese reception right outside where you'll have a chance to talk with our speaker and we can talk amongst ourselves. But now, for the reason why we're here today, and that's to hear from Dr. Huda Zoghbi, who's professor of, get all this, pediatrics, neurology, neuroscience, and molecular and human genetics at Baylor College of Medicine, and in addition is a Howard Hughes Medical Institute investigator. And for those of you who know what that means, it's an extremely impressive set of positions that she held. She received her bachelor's degree from the Beirut Medical School in Lebanon and then moved on to receive her medical degree from Meharry Medical College in Nashville, Tennessee. Her postgraduate training included residencies in pediatrics and pediatric neurology and a research fellowship in molecular and genetics and human genetics at Baylor College of Medicine. Her interests span from neurodevelopment to neurodegeneration. Her discovery that SCA1 is caused by expansion of a polyglutamine tract and that such expansion leads to protein misfolding has profound ramifications, as many late-onset neurological disorders involve the accumulation of mutant proteins in neurons. And I actually understand about half of that. Zoghbi's work in neurodevelopment led to the discovery of the gene MATH1, which governs the development of several components of proprioceptive hearing and vestibular pathways, including cerebellar granule cells and hair cells in the inner ear. Her group also discovered that mutations in MECP2 cause the neurodevelopmental disorder Rett syndrome. We now know that mutations in this gene are responsible for a broad spectrum of disorders, ranging from mild mental retardation to autism. Zoghbi's group generated a mouse model that faithfully replicates most of the behavioral and motor features of human Rett. And they recently showed that MECP2 also regulates RNA splicing in addition to regulating gene expression. Dr. Zoghbi is a member of many professional organizations and serves on the editorial board of several prominent journals. Among her honors are the Sidney Carter Award from the American Academy of Neurology, the Derek Denny Brown Neurological Scholar Award from the American Neurological Association, the E. Mead Johnson Award from the Society of Pediatric Research, the Bernard Sachs Award from the Child Neurology Society, the IPSEN Prize in Neuronal Plasticity, and the well-known Javits Award from the National Institutes of Health. She's also a member of the Institute of Medicine and of the National Academy of Sciences. So as always, let me ask you to silence your pagers, your cell phones, your dogs, and anything else you might have with you that is capable of making noise, and tune into Huda Zoghbi, whose title is Mouse Studies and What They Teach Us About Neurodegeneration, Deafness, and Breathing. Dr. Huda Zoghbi.
Thank you, Jim, for this very generous and kind introduction, and I really hope you'll enjoy this lecture today. So, this lecture really captures the relationship between animals and humans. And what I chose to talk about today is two topics that we study in the laboratory, and left the other one out because about a couple of months ago spoke about that at another lecture at Rice. So what I'd like to tell you today are two stories. Both affect the nervous system. And we did not know initially that both stories will be very important for neurodegeneration. We thought one of them was such, but it turns out both of them are important for neurodegeneration. And the story started in totally different path, and you'll see that. But the key component that really played the major role that helps us in understanding human disease are the mice. So you're going to hear a lot about mice, but you'll hear about them in two different ways. In one way, you'll hear how when we start with a human disease that's devastating, how do we gain an understanding about this disease by studying mice? And the other story you're going to hear about how when we start with a gene that we know very little about, this is making too much noise. Yeah. How we can use the mice to learn really what this gene does, and how that unexpectedly proves to be very relevant to many human ills. So I will try to walk you through the two stories, and I'll tell you exactly how we went about each of them. So the first story involves neurodegeneration. I think almost everyone in this room has heard of the terrible neurodegenerative diseases: Alzheimer's disease, Parkinson's disease, Lou Gehrig disease, or ALS for short. But it's very challenging to study these diseases because, as common as they are, the majority of them are sporadic, which means there'll be one individual in a family, at most maybe two, but not directly related, who might have Alzheimer's or Parkinson. And focus down further. And it's very hard to get at what molecules are making that individual susceptible, what's really going on, why does it take 80 years for that individual to develop Alzheimer's. So it's often the inherited neurodegenerative diseases that are much more helpful to identify molecules that are important for the health of the brain and the integrity of the neurons. And although the inherited diseases, thankfully, are much rarer than the sporadic diseases, at least what we learn from them we're discovering is applicable to the more common diseases. And this is one of the stories I'm going to tell you about today because it started with a human disease. I am a neurologist by training, and part of the reason I decided to go into research is because after you spend enough time in a neurology clinic, you will see there are many devastating disorders that you really, in 1985, when I was in training, you cannot do anything about. And when you see these are happening in some families, as rare as they are, they're devastating, you decide, well, maybe if I did research, I can do something about it. So the story you're going to hear about today is a story of a disease that hit a huge family here in Texas. The disease is called spinocerebellar ataxia. And ataxia is total lack of coordination. It's lack of order, if you will. So when a person is ataxic, they lose their balance. And when they're walking, they seem as if they are drunk. Their speech is slurred. And they fall frequently. And anytime they pick a cup of coffee, they're going to spill it. That's ataxia. And what happens is this fine control that usually happens through the cerebellum of the motor control of movement, how much force you put on a movement, that's motor control of balance, not only affect how we walk and how we speak, but eventually it affects how we swallow when we drink, and eventually it also affects the breathing. So these patients that have this disorder have progressive loss of balance, and eventually their swallowing becomes uncoordinated, their speech will be gone, they'll become bedridden, and they will die because they have choking and recurrent infections. They will die about 10 to 20 years after onset of symptoms. And it's a dominant disease, which means if a person is affected, each of their children has a 50-50 chance of having the disease. And worse yet, it doesn't strike till somebody is 40 or 50 years of age in most of the cases, so most people have kids before they ever know they're affected. So with that, I identified many patients in a town north of here. I don't know how many of you have been to Montgomery, Texas, but it is close to Lake Conroe. It's a beautiful small town, but sadly, this town has a lot of people. If you drive again through it, you might see wheelchairs and canes on the front porches because of all the affected individuals. And this is a diagram of a family that has 7 generations, of which every filled circle and square are an affected, the circle is a female and the square is a male, is an affected individual. And every slash through any of these circles and squares means that person has died from the disease. So you'll see that this is a terrible disease. But what's also unique about this disease when we were studying it in the late 80s was the fact when I talked to the family members, I learned that the people in the first 2 generations had the disease when they were 70 years of age, and 10 years or 15 years later, they died from it. So they lived a normal lifespan, 85, 90 years old. But those that were in the 3rd and 4th generation had disease at 40 or 50 years of age, and those individuals died in their 60s. And so on and so forth. Those that had it at 30 years maybe lived till they were 50. But what we noticed that in the last 2 generations, they were kids. These kids had disease at 4 years of age, and they were dead by 10. So when you now realize that you've had in the same family somebody affected at 4 and somebody affected at 70, this phenomena, which used to be called in the clinical neurology literature anticipation, because you anticipate you're going to find affected people, is not really due to our own anticipation that we can diagnose them earlier. This anticipation is really because that disease somehow is getting worse as it passes from one generation to generation. There's no way somebody can die by 10 years of age because we anticipated the diagnosis. It's because the disease is so much more severe. And it was that phenomena back in 1993, coupled with the finding that Fragile X syndrome, another disease totally unrelated, was found to be caused by what we call today a dynamic mutation, that clued us in that this disease might be caused by a dynamic mutation. And I'll explain to you in a minute what I mean by that, but now you can imagine if a mutation is dynamic, which means a mutation when it passes from father to son to granddaughter, that mutation is changing, and when it changes and something continues to go wrong with the DNA, it can cause a worse disease. And that truly proved to be the case. We looked for a dynamic mutation in this disease and we found it. So what is the dynamic mutation? What the dynamic mutation turned out to be is in our DNA, everybody knows we have 4 DNA bases, cytosine, adenine, guanine, and thymine. And basically, those 4 bases are the alphabet of our genome. You put different sequences, you make different words. These words are really proteins and RNA that does all the function. And it turns out in the gene that's mutated in this family, and families like it, we found, there are 3 base pairs, C, A, and G, that typically, in most of us in this room, range between 6 and 35. The most common one is about 30, 32. So all of us have it variable. We call that polymorphic. That's variation. All of us have variations in our DNA sequence, and whether we have 6 or 10 or 25, we're safe. But it's when we get to the larger numbers, such as 40 or 50 or 60, that's toxic to the neurons. And that's what happened in this family. This mutation expanded, this repetitive sequence got bigger, and if you take a Shakespearean poem and take one word and repeat it, the poem will be destroyed. And that's really what's happening here, if you will. That protein now is not the same protein. And what we discovered is that those individuals that had disease in their 70s had 40 repeats, but the youngest one had 70. So the bigger the repeat, the worse is the disease. And this repeats turned to be present inside the part of the gene that makes a protein. This was the very first ataxia gene ever cloned. So there are now, we know, about 30 or so different inherited balance disorders or ataxias. And this was the first one, so we didn't know what to call the protein. We called it ataxin 1, being the first ataxia gene. And it turned out this 3 base pairs encode for a specific amino acid called glutamine, and that glutamine, as I mentioned, gets longer in the patients. And I just want to mention here that today we know there are 9 different diseases caused by a similar mechanism, which means the mechanism is expansion of these 3 bases of DNA in different genes. So these 9 diseases, each of them is caused by a different gene, and many of them cause balance disorders. You'll see there's SCA1, this is the disease we studied, another SCA6, SCA2, SCA3. We're up to SCA17, but the genes have been only known for a few of them, and here are some examples. But probably many of you recognize the most famous one, Huntington disease. It's also the same kind of mechanism. And in each of those, different gene expressed in all cells, but different neurons degenerate in each of these diseases. So here it's one type of neuron, and in the ataxias, it's usually this beautiful neuron called the Purkinje cells, which you'll see images of it. So now that you found a gene, you can help these families because you can diagnose them. You can tell somebody who's 30, if they wish to know, and they're properly counseled, that they carry the gene mutation, and although they are not symptomatic yet, at least they can make their own decision whether they wish to have kids or adopt kids or choose whatever method they want to prevent the recurrence of the disease of the family. But most importantly, for those that are affected, what can you do? How can you study why is it that many of us in this room that have 35 glutamines in this protein, we have these very nice and healthy Purkinje cells, versus those individuals that just have 40, they lose these Purkinje cells. Why is it? Just a slight change in the number of amino acids. And this is where mice come in handy. What we have found is if we put the human mutation in mice, you can create the same balance problem as we've seen in the humans. So these mice, this particular one that's falling, that's having balance problem, this mouse is falling on a padded surface, is having difficulty maintaining balance because we only put in its Purkinje cell, it just doesn't have the coordination to hold on. We put in its Purkinje cell the human gene. We took the human gene with the long repeat, and we put it in just in one cell type. We put it in the one cell. We didn't put it where it's expressed everywhere. I'll show you another mouse about that. But that's told us that Purkinje cell is really important. That's the cell that's hurting the most in this disease, and this mouse has the disorders. And this is a real image now of the Purkinje cells. And that's the value of having mouse models, is now you can look at as much tissue as you want, because although these human patients pass away, it's not always that you get good quality tissue. So if you look at a slice of the cerebellum, the part of the brain that controls balance and coordination, you'll find these beautiful Purkinje cells stained here with specific marker, and you'll see all these very intricate arbors for each, the dendrites for each of these cells. And you'll notice in the mice that express this protein, the Purkinje cells, many of the cells are gone, and we lost their arbors. And the other advantage of having mice is you can start studying them before they're even symptomatic. So in humans, if you get tissue, it's after the patient suffered the long course of the disease. But in mice, you can begin to look at different steps to see exactly what happens in every neuron at the different steps of the disease. And when we did that, we noticed something very peculiar. This is the Purkinje cell now staining the nucleus of the Purkinje cell with an antibody to ataxin 1. So the cell that have ataxin 1 will be brown. And in normal mouse, you'll see these cells nicely staining. Here you'll see when you have that long glutamine repeat in the protein, it did something different to the protein. It made the protein accumulate in these dark spots, these dark circles. And we went back now and looked at tissue from one of our patients who died from this disease. And this is one of our patients who had the disease at 4 and died at 10. And when we look into her neurons, we found this dark staining material, which is ataxin 1, among many other things, in the nuclei of the neurons. And knowing that in the patient, unlike the mouse, we didn't put extra copies, it's just the one copy that's mutated, we figured, well, the patient doesn't have extra copies. So the fact it's accumulating usually could be either because you make too much or you cannot clear it, because proteins normally are cleared in the cell. And usually to clear a protein, you have to put a flag on it with this molecule, ubiquitin. You tag it with this molecule, ubiquitin, and then the protein is degraded. And what we noticed that all these aggregating ataxin-1 molecules have a lot of ubiquitin on them. And that clued us in that perhaps when you now put a long glutamine tract in a protein, the protein perhaps changes its shape. That gave us the first clue. And when it changes its shape, maybe it cannot be degraded. And I think I have a nice animation that sort of illustrates this. So imagine this protein ataxin-1 being that yellow that yellow molecule here. There is an enzyme which carries ubiquitin. The pink little balls are ubiquitin. And this is ataxin. So imagine this is normal ataxin. There will be a series of enzymatic reactions that will ligate the ubiquitin onto a protein. This happens for many proteins in our cells. These enzymes put ubiquitin. Whenever a protein does its function, it's supposed to be degraded, then a new amount is made, and then it's degraded. So now that ataxin-1 has many ubiquitin molecules, it goes through this big, giant structure in the nuclei, and the cap opens, the protein, you can see it unfolding so that it can get into the part that chops the protein into pieces. That's called the catalytic core of the proteasome, and the protein is degraded. So we knew that this is how ataxin-1 is normally degraded in the cell, by this ubiquitin proteasome system. And now we wanted to see what happens to the mutant protein. So imagine this is being mutant ataxin-1 that takes on a different shape, different form. The cell recognizes it, can ubiquitinate it. It brings it to the proteasome to be degraded. The cap will be open, but it's stuck. It just doesn't quite unfold to get in and be degraded. And this is what's happening in this particular case. And I'm not showing you all the biochemical data. You don't want to see all that, but this sums it up. So having discovered that, this brings me back to my original point, that is there a relationship between this very rare neurodegenerative disease you've never heard about, probably most of you in this room, and the more common neurodegenerative diseases? And the answer is yes. So what you see on this slide, you've seen brain sections from my patient with SCA1, but now you'll notice that this feature of having accumulating proteins is common to Huntington disease. It's common to Parkinson's disease. In Parkinson, we see this accumulation of another protein. It's always a different protein, but the idea is the same. These neurons are having to deal with a protein that's misfolding, that's resisting clearance. You see it in Lou Gehrig disease, ALS. This has been always noted that in prion diseases, we have these proteins, prion proteins, that take on a different form and they resist degradation. And we also are aware of some accumulations of proteins such as amyloid plaque and tangles in Alzheimer's disease. So now we sort of began to really pinpoint a unifying theme in many of the neurodegenerative disorders, and this unifying theme is that there is a misfolding of certain proteins, and certain mutations in certain genes cause that. I just wanted to show you one other way one can use mice, and this is genetic engineering. It's slightly more complicated than the first mouse. The first mouse, we basically just took the human gene, took the DNA, injected it in the fertilized egg, and we got a mouse that have an extra copy of the human gene. But one can do even more than that. One can now make the mouse gene look like a human gene. And what I mean by that, what you can do now, go into the mouse genome, insert the long repeats, like our human patients have it, and leave the DNA intact. That's the only change you do to the DNA, which means you humanize the mouse DNA to have the mutation expressed at the same level as it is in the human, and to have it in every neuron. And this is the case here. I just want to show you that the mouse normally has 2 glutamines, and when we can genetically engineer it to put in the long repeat, we get a mouse that pretty much reproduces now all the features of SCA1. The first mouse only had the balance problem because we only put it in one neuron. But now if you put it in all the neurons, because you just modified the mouse gene, you get all the symptoms of the human disease, the ataxia, spasticity, some memory problems, and the premature death. So this mouse model, which you see a picture of this mouse now, which is a lot more severe because this mouse now expresses this protein in all of its neurons, you can see how it's losing a lot of muscles eventually and has some problems with spines because of the weakness. And I'm just going to maybe move through a couple of slides here just to show you that for another SCA7 that we've studied in the lab, this one has retinal degeneration. And in this particular case, what we did, this is such a severe disease that sometimes we have children and babies affected with it. You have such a huge repeat in this particular disease that you might have infantile onset. So when we put the same size repeat that has been seen in a human patient that had onset at 1 month and developed, this disease affects the eyes and causes seizures and death by 6 months, we saw exactly the same age of onset in the mice and the same approximate age of death. And the point I want to make here is that these models really help you, are very authentic models, and you can use them quite a bit to study disease pathogenesis. And one thing about this mouse, I don't know what's happening to the slides here, but this is the SCA7 mouse that has also an eye phenotype. So you can see this mouse is different because it has degeneration of the photoreceptors in the eyes. And one thing that was very important from the study of this mouse model, I mentioned to you that what we think the glutamine expansion is doing is causing the protein to accumulate. And what you'll see here, that in healthy mice, you hardly detect this protein. But in the SCA7 mouse, if you follow this mouse over its whole lifespan, you'll see the protein very gradually accumulating. And this really provided us in vivo evidence, as we would say, that the glutamine expansion is really affecting the stability of the protein and causing it to accumulate. So now that we've pretty much pinpointed that it is about protein folding and accumulation for this class of diseases, what can we do about it? And we know that in the cells, in addition to the machines that degrade proteins, there are other machinery that helps refold proteins that take on a bad form or a bad shape. We call them misfolded proteins. And usually these chaperones will either help alter the shape of ataxin-1 to help it be shredded now through the proteasome, or maybe give it a slightly better shape. And we wanted to test that, and that's again where mice can be helpful. We took our SCA1 mice that you just saw them falling on the rotating rod because of their balance problem, and mated them with mice that make extra copies of chaperones. You can put extra genes in mice. So here we put extra copy of this protein that we know can help refold protein. And now you got a mouse that has both genes. And if you now put this mouse on a rotating rod, and I just want to show you the data rather than, again, balancing themselves, you'll see that the SCA1 mouse will learn how to stay longer if you train them, but they will fall at about 300 seconds. However, if they have extra copies of chaperones, they can now last longer on the rotarod. And when we look at the brain pathology, it looks better. So this was really important because it told us, okay, this is one molecule, one protein that we know can help this class of disease. But given that protein misfolding is a problem for many neurodegenerative diseases, many other investigators have done similar studies. About half a dozen of the neurodegenerative diseases has been studied as such, and chaperones suppress the neurodegeneration in most of these. So this provides a molecular target, and if one can now generate a chemical chaperone, a pill that can behave in the cell, can cross the blood-brain barrier and behave as such, that would be a great advance. And I think there are many labs and companies working on that. Let's see here. I think that I'm going to skip through this one and just show you one other pathway we've identified about ataxin-1. We discovered that ataxin-1, many proteins can be modified. You basically add a little chemical, in this case a phosphate group, or change them or cleave them. They can have different shapes and forms. And what's nice about this story here we found with ataxin-1 is that this enzyme, a kinase, is what adds this phosphate group to ataxin-1. And what we discovered through biochemical and genetic studies, that when you have the protein phosphorylated, it tends to accumulate more. It tends to take on the misfolded shape more, especially when it has a long glutamine repeat. And why is this important? Why I'm mentioning this story? Because this is being an enzyme, there are many chemicals that can inhibit enzymes. And there are chemicals that inhibit this specific pathway. So in this particular case, we're using our mice to try what we call preclinical studies. And what we're doing here is using the SCA1 knock-in mice as well as the transgenic mice that replicate features of the disease and giving them compounds that inhibit this enzyme, and we're trying to see would this help them. And these studies take a long time, but at least if you do them in animal models and if you feel you see an improvement, then at least you can optimize the drugs to hopefully go to human patients. So one last part of the story on SCA1 that I'd like to tell you that I think is very important, and I hope you will appreciate the implication of this study. So we got a human degenerative disease, a disease that takes decades to take shape, and right now we don't have a treatment. Let's say 3 years from now there is a great drug. Are all these individuals who are affected, are they going to benefit from that, or are we only going to help people when they're not symptomatic? We did not know the answer to this, but with mice, you can know the answer to this. And how can you know the answer to this? You can essentially engineer the human mutation, the gene with the human mutation, and you put it back in the mouse, and you do a little genetic engineering trick so that you can have the gene either on or off on demand. You can turn the human mutation gene on, and the mouse, just like the first one you saw falling, is going to develop neurodegeneration. And if you decide, well, I'm going to let this mouse get neurodegeneration for 3 months, but now I'm going to turn it off and see what happens, you can watch and see what happens, because now the gene is not expressed anymore. And the way one can do this trick is by using specific DNA sequences that respond to a drug called doxycycline, which is an antibiotic. So the antibiotic does not treat the disease. What it really does is just help the gene be on or off. So what I want to show you here is what happens when you do that. Basically, in these animals here, we had the gene. This is now, again, the rotarod. We use the rotarod because it's a very easy way. You put the mice, you can measure how long, you can quantify the data. So we use that quite a bit, and you'll see different graphs for that. But what you'll notice here, if the gene has been on for 8 weeks, the animals do okay. If it's been on for 6 weeks and then off for 2 weeks, so we're measuring them, both animals are 8 weeks old, but you don't see much difference. When it's been on for 6 weeks and off for 2 weeks, you don't see much difference. However, if you now keep the mutant gene on for 10 weeks, so these poor animals have been exposed to the human mutation for 10 weeks, whereas these have been exposed for 6 weeks and then we turned it off for 4 weeks, you see a slight difference. But let's watch now if we keep, remember, this is a late onset progressive degenerative disease. What will happen if we keep it on for 12 weeks, which means these animals exposed to the mutation for 3 months, versus animals that had it 6 weeks on and 6 weeks off? You'll see a huge difference. And what will happen here is that if you compare this to healthy mice, the 12-week, this is the curve for healthy mice. What this is telling us is that even after being exposed to the human mutation for 6 weeks, if you stop it, you can reverse the disease. And a 6 weeks old mouse is a teenager. It's a young adult, sort of, just to give you an idea about the age here. So this was quite an important discovery. And if you were to look at the pathology now, here's the Purkinje cells, and you can see there's a lot lost when the gene has been on for 24 weeks. And here it is when the gene has been on 12 weeks, off 12 weeks. Even if you kept it on for 3 months, you still see massive recovery. And this is a summary of the data we've done on the recovery. If you turn it off quite early, after 6 weeks, you get complete recovery. After 12 weeks, you get almost complete recovery. But even if you let it go on for beyond 6 months, you get quite a bit of recovery. And this was a very exciting finding, because what this is telling us that these protein misfolding disorders, if we can just help the neurons a little bit and somehow figure a way to decrease the burden of that toxic protein, we have a chance of recovery. So to sum up this first half of the talk, which is the story that started with human patients, I hope I convinced you that the studies of mouse models helped us really understand the importance of protein accumulation and folding in neurodegeneration. And I didn't go through all the cross-species studies, but at least I mentioned to you the AKT kinase pathway that led us now to do preclinical trials. But most importantly, we have two facts going for us in neurodegenerative diseases. One, they take years and decades to show up. And second, now we know they can be reversed. So I think any drug treatments we can develop over the next few years that even if it slightly decreases the burden of the toxic protein will probably have quite a remarkable recovery, and we may help decrease the burden of disease and give patients perhaps an additional 10 years of higher quality life. And I think that's quite exciting. I did not mention everybody who's done all the work, but I would like to mention that it's always wonderful graduate students and fellows in the lab that do all the stories, and I have had a long-term collaboration with Harry Orr at the University of Minnesota and Juan Botas at Baylor, and we're grateful for funding from NIH and HHMI. So I think I'm going to now switch to this other story, and even the color of the slide changes, just to just to sort of get you refreshed. So for this story, it is really totally different. This is about a big word, proprioception. What is proprioception? The best way for me to explain to you proprioception is to give you examples. If you were to close your eyes right now, you will still know where your right arm is, where your left leg is, exactly where is your body with respect to the chair. You'll know all of that. And the reason you know that, you have a sort of a sixth sense. You're getting some sensory feedback to your brain where you know your whereabouts. When you get up in the middle of the night to get a drink of water, you don't have to turn the lights on. You pretty much know where to go, how to come back, and that's because of your sense of proprioception. So the first part of the talk was about motor control of balance, the output from the cerebellum that helps you decide how much force to put on a cup of water. But for that output to happen, there has to be a sensory input. You have to sort of know when you move your arm, where is your arm going to meet the cup? And you're going to have to know exactly the position of your arm. So the two really work together to give you that sense. And as I mentioned, Charles Bell called it the sixth sense. It's because of that, because it happens subconsciously. You just do it automatically. You don't think when you're going down the steps. You don't really look at your feet, because once you know the proportion of the steps in your house, that's it. If you're ever at a place where the steps are the same proportion and one step off, you'll often fall. That happened to me, and I almost broke a leg. That's because of proprioception, because we don't think about it. So, and it's an indispensable sense. And I'm going to give you a case history to tell you about how we know it's indispensable. This is a story that was written in a book published by MIT Press called A Pride and a Daily Marathon. And it's a story of a man who suffered a viral infection that wiped out the neurons in his peripheral nervous system that mediate the sense of proprioception. And if you read the story of this man, it's very devastating, because he describes himself as a heap in bed that could do nothing. Just nothing. He's just like a piece of meat, he says. He was embarrassed when nurses came to give him his medications. If he tries to get up, he'll be hitting them, because he had no clue where his arms and legs and so on were. And he finally decided to use conscious effort to control his movements. And after 10 months, he taught himself to take a few steps, using visual cues and really using a lot of mental concentration to get that going. And hence the name of the book, Pride and a Daily Marathon, because taking a few steps was a daily marathon for him. So this was a viral infection, and of course from that you can't learn much about the circuitry, all the neurons that talk to each other to do proprioception. And I was interested in proprioception, because with my interest in balance disorders, I was interested in the whole story. But you don't find human patients, because they don't survive. It's really very hard to live if you lack total proprioception. So how do you go about it? Well, the way we went about it, we're going to talk about one more animal in this slide, Drosophila, the fruit fly. The fruit fly, Drosophila, has a gene that's called atonal that specifies all these things shown in yellow, which are actually the proprioceptive organs in the fly. This is how the fly knows where its limbs are, where its wings, and it also uses them as hearing organs. And that gene is called atonal. It's a gene that is very important to specify all these receptors. The flies are called atonal because without it, they have no tone and they can't coordinate their movements and they're deaf. So we decided, well, since flies usually don't have too many genes to take care of things. They have one gene typically for one important function. Since we knew this gene was so important in the fruit fly, we made the hypothesis that maybe if found the mouse homologue, maybe that gene will be important in mice and possibly humans. And there are molecular ways you can isolate a mouse homologue of a gene, and we called it mouse atonal homologue 1, or MATH1 for short. So when we obtained MATH1, we went now to the mice, and one thing I should mention, just like you can put a human mutation in a mouse, you can also engineer a mouse to lack a gene. So what we did is we engineered a mouse that lacks the MATH1 gene, but at the same time, we made it such that wherever MATH1 is expressed, the cells will be labeled blue. So this way we can catch where this gene is normally expressed by adding a special chemical due to a gene called LAC-Z. So what we found out during development, that this gene is expressed in what we call the external granule cells, and we found out that the mice that lack this gene lack these cells, and these cells make up millions and millions of neurons in the cerebellum that are important for proprioception. Then we started looking where else is this gene expressed, and you can see these nice little blue spots, and you can see these joints staining blue, and this is altered when the mouse lack the MATH1 gene. What are these? The little dots are Merkel cells. They are the ones that making you feel your back or when you have pressure on your skin. These are mechanoreceptors that if you touch your skin, that sensory pressure gets transmitted via the spinal cord and then to the brain via these cells. So they talk to neurons in the central nervous system. And this mouse lacked those as well. Then we continued looking in this mouse, and to our surprise, we found that there are few blue cells in the inner ear. Here's the inner ear where we have the cochlea. The cochlea is the hearing organ in the ear. And then there's these 3 canals here, part of the vestibular system. And the vestibular system has little mechanoreceptors in it that we call hair cells, that typically when you move your head, the fluid in the inner ear will touch these hair cells, and that movements will send a signal now to the brain to tell the brain where, if you're tilting your head to the left or to the right, it essentially helps you know the position of your head. And of course, the cochlear hair cells are receiving the energy, the sound signal, and that the sound signal causes, again, movement of these cells which talk to neurons, and that mediates hearing. So when we saw that expression, we decided to see what happens when you don't have MATH1, what happens in the inner ears when there is no MATH1. I'm just going to show you one slide here. This is an electron microscopy picture that shows you these beautiful hair cells. They look like bundles in the cochlea, and they're all gone in the MATH1 mice where the gene has been gone. So what this told us, that this gene is essential and necessary to make the inner ear hair cells. Without this gene, you cannot tell the balance of the head and you cannot hear. And I just want to tell you a story that was taken up then by the Yehosh Raphael at the University of Michigan. What Yehosh did, he is actually an inner ear hair cell biologist. He was very interested to know, well, the Zoghbi group showed that you need MATH1 to make inner ear hair cells. What I want to see, is MATH1 sufficient to make hair cells? So what he did, he took guinea pigs' ears and he damaged all the hair cells, and then he put MATH1. And when he did that, he found now the guinea pigs made more new hair cells. So it's not only necessary whereby when you delete it, you lose the inner ear hair cells. It's also sufficient. If you damage hair cells and you put it back, you make new inner ear hair cells. And what was really, this was a paper he published 2 years ago, but his latest paper, which he just published in Nature Medicine, was very, very exciting Because what he did, he took the guinea pig, he damaged their hair cells, he did hearing tests on them physiologically and showed that they are deaf, and then put MATH1 using local gene therapy in the inner ear, and found that not only do they make hair cells, but the animals can hear. He went back and recorded, and these animals can hear. So this is really a very exciting result because it tells us that this is local. He used adenovirus. These are not the same type of gene therapy vectors that has caused some alarming things by integrating in the genome. These are different types. They're topical, really easy access. All we have to do now is get this to work in a primate. This all worked in rodents. And we know that the human homologue of MATH1 exists, and it's highly conserved. The only challenge is the cochlea in a primate is slightly different than the cochlea in the guinea pig. So I just want to put a spin of reality here that it's not as easy to get to the cochlea in a primate. And Yehosh is working on that. And in fact, we're trying to recruit him here to Houston, to Baylor, because it'll be great to have such an investigator doing applied translational research. So what we learned from studying MATH1 was that it's, as I mentioned, necessary for cochlea and balance organ, for these neurons in the cerebellum, for another set of neurons in the spinal cord. And really what's common with all these neurons is that three of them belong to the proprioceptive pathway. So notice that this is what this gene does in the fly, and it seems to be doing the same thing in the mouse. But what was also notable about these mice, if you don't have MATH1, the mice die at birth. They never took a breath. So we wanted to pursue further what does MATH1 do. And while we were doing that, we were shocked to find it expressed in the intestine. We couldn't figure out what MATH1 is doing in the intestine. Here's the gene important for hearing and balance. What business does it have in the intestine? And so I had to learn about the intestine for a few years, and it turned out to be a really fun story. So you have to hear about it. I discovered, which I didn't know, that the intestine has 4 types of cells. There are a lot of cells that are these clear cells are the enterocytes. These are the cells when you eat are helping you absorb all the nutrients in the foods, and these are the majority of the cells. But then there are 3 other types of cells. One is called an endocrine cell, one is called PANF, and one is called a mucus-secreting cell. And that's this one here. And these 3 types of cells are all secretory. The endocrine cells makes about 16 different, there's 16 different of them. They make different peptides that helps us move food along. The mucus keeps the colon and the intestine with a nice amount of mucus against to help food and bulk along. And the PANF cell that sits down here is basically an antimicrobial cell. It secretes peptides that attack bacteria and germs. So there are 3 types of cells. They're secretory, and then there is these enterocytes. And all of them, oops, what happened? Let's see. No, everything went, now it's back. Okay, good. So what was known at the time was that, as I just mentioned to you, there was known that these 4 types of cells and what they do, but nothing is known. How does I wanted to show you that there's a stem cell in the gut, and it's this stem cell that sits here, divides, and gives rise to all of those cells. So we keep making these cells new every 3 weeks. So the gut, we're always replenishing the cells. So that was known. But what was not known is how can a stem cell decide to go from this one stem cell to give all these types? And that's what we discovered by studying MATH1. We found out that the MATH1 knockout mice lack all the secretory cells. So the one that make mucus, that are stained here in blue, were lacking. The one that make all the endocrine hormones in the gut, in the intestine, were lacking. And again, I'm not going to show you a lot of the data, but I'd like to show you a summary slide, what we've learned from studying MATH1 and things that work downstream from MATH1. Here's again another picture. As we got smarter, as we learned about the gut, we can now put things in better color. So here's the stem cell. We discovered that MATH1 is expressed in one of the daughters of the stem cell, when this daughter can continue to divide. So basically, cells that come down from the MAF1 cell gives us all these 3 types of secretory cells. Another gene, another group discovered gives us the enterocytes. And we recently found how cells decide between these fates and so on and so forth. Why is this important? It's important because you actually need, for gut homeostasis in health, you need to keep replenishing them, so you need to have this transcription factor always helping maintaining that. And what's really, really interesting is that these endocrine cells that are dependent on MAF1, they signal back to the nervous system of the gut and then help the gut feel the movement and control and coordinated movement. So it sort of really did bring back MAF1 to gut proprioception, but indirectly via the enteroendocrine system, which was a total surprise. Finally, we wanted to know, why do these animals lack breathing? That's the last part. We knew these animals are born alive, but they don't breathe, so they die. And I mentioned to you, this is a mouse embryo, that when we delete this gene, all the cells that normally express it will stain blue. And there's this structure in the back of the brain during development that's called the rhombic clip, because it looks like a rhombus. And a lot of cells are derived from that rhombic clip. And we were able to define many cell types that derive from that. This is the work of a graduate student, and I'm just going to show you actually the summary slide here in the interest of time. I'll show you just the summary slide. I know it's a busy slide, but it turned out, here's the rhombic clip. We discovered many neurons that migrate from the rhombic clip, shown in this way. This is the part of the hindbrain where all this is happening. And they migrate at different times, and they specify different neurons in the nervous system. And to cut a long story short, let me tell you what all the neurons that depend on MAF1. Many neurons in the cerebellum, the cochlear nucleus neurons, vestibular nucleus neurons. Now remember, it's the inner ear hair cells that are for hearing. So it turns out that MAF1 specify the outside mechanoreceptor, the hair cell, the cochlear nucleus, which is in the brainstem, and in the next set of nucleus that synapse with that. And the same for the vestibular system, the same for the proprioceptive system. So, in fact, it is amazing that the same gene that control hearing and proprioception in the fruit fly is highly conserved in the mammal to control exactly the same function, hearing and balance and coordination. And while in the fruit fly there's just a simple organ called the chordotonal organ to do all this, in mammals, there are multiple neurons that have to talk to each other to do this, and MAF1 specifies each one of them. And what we're doing right now is we identified many candidate neurons for the regulation of breathing, but what we need to do is we have to pinpoint these neurons, and this is just to show you how the animals look when they don't have MAF1. Their lungs don't inflate. And when we do physiology on them, we know that they don't breathe normally. We can record, and this is in collaboration with John Green. We know that they do not maintain a respiratory rhythm. And what we're doing right now, we're trying to pinpoint exactly which of these neurons are really essential by deleting them. Now we made a special mouse where we can knock out MAF1 in just a subset of neurons each time. And once we find the neurons where the animals lose breathing, then we can say these are the neurons important for breathing. And why are we interested in this? We're interested because sudden infant death syndrome is a perplexing disorder, and nobody really knows about all the neurons that control breathing and modulate breathing. So this is the hindbrain. This is the rhombic clip here. And these are what we call rhombomeres that give rise to all these different neurons. And we know that we can knock it out at one region at a time, so depending on which specific enzyme we use to delete MAF1. So you can see in black we can delete it here or here or here or here, and that will allow us to pinpoint the neurons responsible for breathing. So this sums up the story on MAF1, a gene that's essential for the development of multiple components of balance, coordination, vestibular function, hearing, and the whole sense of proprioception and breathing, and we hope to identify those breathing neurons. This is work done by many people, again in the lab, who contributed to different stories. Melissa worked on the gut, and these two fellows initially characterized the mutants, and a graduate student and a postdoc now are trying to pinpoint the breathing neurons along with other collaborators. Thank you.
Thank you very much, Huda. I think I can speak for all of us in the room who like to breathe and hear. That was fascinating. We now have some time for questions. Steve Kleinberg is over on that side of the room with a microphone. I've got a microphone on this side. So we ask you to raise your hands, and if you ask a question, ask it loudly and clearly enough so that Dr. Zoghbi doesn't need to repeat it. Have one? I could start by asking one. We've heard an awful lot over the last 5 years about the importance of stem cell research for solving neurodegenerative diseases and so forth. Do you think it's possible that techniques like the ones that you're describing could produce the hoped-for cure without the need for some of the more controversial methods?
Right. So I think that's really an important question. And the question, can you find adult stem cells that perhaps can do the work? There are two types of stem cells. There are the embryonic stem cells, and the embryonic stem cells really have the potential to become anything you want them to be. Neuron, pancreatic cell, what have you. And I think that's the beauty of them, and they can last a long time doing that, keep replenishing themselves. The challenge, of course, with those cells is availability of quality cells, and then getting them for nervous system disorders. The challenge is not only getting them into the brain, but getting them to wire properly. So I think there's probably, at some point in time, there will be some benefit from the use of such cells for neurodegenerative diseases, but I think we have many obstacles to overcome. Now, in other organs, I think it's much easier. If you think about the pancreas or the gut, the delivery to such organs is easier, but more importantly, I think all they have to do is just go to that spot. They don't have to have the beautiful connections and architecture that neurons have to make in the brain to function properly. So my hope is that at least we can use stem cells for diseases like diabetes, perhaps inflammatory disorders of the colon or some other peripheral diseases, and hopefully in between we can figure out how to put neurons properly so that they wire properly. Now, adult stem cells, how much are these useful? I don't think we know yet. For sure, for hematopoietic stem cells in the blood and maybe in the gut, those will have some use. But whether those will have the same efficacy as the embryonic stem cells that can last a lifetime and can be differentiated into many things, it remains to be seen.
I had a question. I was just wondering if proprioception is a separate sense or if it's just a combination of like balance and touch. Because like when you said when you had a mutant for MATH1, you lost the mechanoreceptors and also the hairs in the ear, and so it seems like that's proprioception.
Right.
Or is it different?
No, hearing is not proprioception, but proprioception is really different from touch because proprioception is due to movement and position. It's position sense. That's the best way I can describe it. And the normal touch is mediated by different receptors than really having some pressure touch that affects those Merkel cells and the mechanoreceptors. So in proprioception, the sensation happens due to movement that affects some mechanoreceptors in the joints. So for example, if you close your eye, and if I touch your finger, you'll know I touched it. That's not proprioception. But you'll be able, if your eyes closed, to know if I'm moving your finger up or down. That's proprioception. And if you don't have proprioception, you'll still know I touched you, but you will not know which direction your finger is going. Okay?
Can environmental insults create the glutamate replication and attending problems in otherwise healthy subjects?
So the glutamine expansion is not an environmentally, in general, is not an environmentally induced factor. It's really something that happens in the DNA as a major error. And we don't quite understand how it happens. We know that the upper limit of normal is, say, 35 repeats, and exactly what goes wrong and someone goes from 35 to 36 and then it goes wild and becomes dynamic, we haven't figured it out. Nobody has quite yet. It's conceivable that maybe there is some error in DNA repair enzymes and maybe some damage happens, but we don't have evidence for that. But I thought you were going to ask me a different question, so I'm going to answer it even though you didn't ask it. So that's a genetic disease, not environmental. But I think the more interesting question, can environmental toxins affect protein folding and make us susceptible to neurodegenerative effects? Because as I mentioned, we started by saying Alzheimer, Parkinson, all these are really sporadic. And I think that that's really an important question. I'm sure that there are environmental factors that tax the neurons. So a neuron cannot divide, is equipped with all the degradation machinery, with all the protein folding machinery that it needs, but it cannot divide. Whatever it has to do it, most neurons at least, have to do it for a lifetime, 80 years or 90 years. So basically, you can imagine if you're really taxing these neurons, whether it's through being exposed to carbon monoxide or whether it is being exposed to too much of a high-fat diet. I'm just saying things, pesticide, whatever toxin we might be exposing these neurons to, I have no doubt that that's probably is causing additional protein misfolding for a bunch of proteins, not necessarily ataxin 1, and that probably contributes to neurodegeneration. So I think that's really an important question, and I don't think we know the extent. We used to think of everything being environmental, then everything became genetic, and I think now we're finally wise, and we know it's both. It's really genes and environment. So let's take two people. One person may have amazing number of chaperone molecules in their cells, no matter what they do. They smoke, they eat high fat, they drink, they still have their balance. Another person, maybe their chaperones, due to a variation in their gene, they're slightly 70% capacity. A little extra alcohol might make them lose their balance when they're 60, or maybe they might get Parkinson's disease just because they sprayed their gardens way too many times with pesticides. So, I mean, we don't know these things, and I think until we understand all of this and the susceptibilities, that's going to be a very important area of research because we can prevent it.
Because one of the hypotheses for Alzheimer's disease is head injury. Trauma. And but it's more than just the area of the trauma because
Correct.
It triggers and spreads. That's why
Yes. In fact, I'm glad you asked that again. Trauma requires the same heat shock proteins as the protein misfolding disorders. So head injury clearly requires, anytime you have trauma, you induce all these heat shock or protein chaperones. The same happens in heat stroke. Really, heat stroke, what is it about? It's about exhausting all the chaperones because you induce them with heat shock, and having a sunstroke exhausts them, and that's why the brain finally gets destroyed. So there are many things that tax that protein folding system. And we have to understand more and more about it so that we can at least avoid these things. And even stress, I wouldn't be surprised that could also throw somebody off. I mean, elderly do not tolerate major stressors, be it heat, be it stroke, trauma, or be it stress, as younger brains. And all of these diseases, really, if you watch a 90-year-old or a 100-year-old person, I should say 90 because Dr. DeBakey looks fabulous, but 100-year-old, 105-year-old person, you will notice that really they have mild symptoms of all these diseases. Their balance, the movement, a little tremor, a little memory. So even the healthy individuals, we do tax the nervous system with aging.
Could I ask, is autism a proprioceptive disease? Because I've heard that an autistic child has said he has to do the flapping so he can tell where his body is.
Yes, that's a very, very interesting question. And it's a question, in fact, we deal with in our lab. We're thinking about, nobody really understands what causes autism yet. But it's absolutely true. Those autistic kids that can communicate, they do express a challenge with knowing where their body parts are, and that's why they continue to flap their arms and move on. And they also have intestinal problems. They have a lot of intestinal problems, either too much activity, too little activity, too many Paneth cells, too little this cell, and so on. So in fact, one thing we contemplated is to look at our mice that lack one copy of MAF1, not two copies. So those that lack one copy are alive and well, to see if they have any social behavior phenotypes. We don't know if this is contributing, but that could be.
One down here, one up there, and then we probably should wait.
Okay.
What's progress, if any, toward a simple and expensive test for that gene replication?
There is a test for all of these now triplet repeat expansions. It's a blood test, basically. One gives a blood test, and they can check the DNA, check the size of the repeat, and one can tell if one carries one of those mutations. So there are 9 diseases that are characterized by this type of mutation, and for all 9 genes, there is an assay.
Huda, you're part of a series that we've been hearing this year in Scientia on the relationship to human beings and animals, and you pointed out in the great use of the rat or the mouse is that it is very similar to us.
Yes.
And yet at the same time totally controllable by us and short-lived. Help us think about what are the moral implications? How do you think about the relationship between us and the mice, and do we have responsibilities? And what happens as you get to chimpanzees who are even more like us?
Yes.
Any thoughts about that as you think about it?
So you're asking me the philosophical side of our research. Yes. No, trust me, I think about it, and you said that we're very similar, and we have control over them. And I just worry that in another life, they're going to have control over me. That's the first thing I think about. I'm going to come back, and they're going to take care of me. But I think that it's really an important question, and Jim and I were talking a little bit about this before. So there are 2 issues I'd like to address. The first issue is whenever you do animal work, you do the work with the lowest possible species that you can do to get answers, and you reserve, at least in my philosophy, you reserve non-human primates for things that you really have to establish in monkeys before you go to humans. But what are the bigger implications of using animals in research? I think that, believe it or not, it's really a two-way street. In one way, you can think about my poor human SCA1 patients being a model for ataxia. And we know there are many natural mouse mutants that have ataxia. We know there are many dogs that have neurologic syndromes that suffer from them, whether it's motor neuron disease, whether it's narcolepsy, whether it's epilepsy. So it really does go both ways. And we tend, as scientists, when we're presenting our work, to focus on how we use animals to understand disease, because we don't work to help cure animal diseases. But if you were to take the Merck animal experts, and you sit on that board and you see what they do, they will tell you all the work that has happened that have cured, heartworm in the dog, or that have cured this epilepsy or that epilepsy. So the big picture for me is that science should be done really for the better of all organisms, if we can. And you use animals when you have to. When the family has epilepsy, you use that family to clone the gene. And that might help you treat an epilepsy in a dog or in a human. And vice versa, you have ataxia, you can model it in a mouse, but eventually you might learn something about that benefit another organism. So I don't know if that answered your question, but that's at least my view on it.
Would you give the same talk to an audience of mice?
Trust me, I have an audience of mice. I talk to them all the time. Hey, we really do. I mean, it is true. You get attached to these creatures, and you really do want to make sure they have the most comfortable cages, they have the most food, bedding, toys, and they are sensitive creatures. You give them more toys, they live better, they do better, you stress them, they do poorly. So I think that being sensitive to all of that is really critical.
We do have cheese out in the foyer. Huda, thank you for a wonderful talk.
Thank you.